biologist at Harvard, and I have a question for you. Imagine you could look really closely inside of your finger. What do you think you might see? Probably some bones, muscle, blood, skin. What if you could look even more closely? If you could zoom in close enough on your finger, you would see that all of those parts of your finger, and actually all parts of your body, are made up of cells. And if you caught a cell at just the right time, you might see something that looks a little like this. What we saw there was a cell dividing, an essential and fascinating part of life. All of us started out as single cells, so in order to get to the large, complex organisms we are today, our cells had to divide over and over. And even when we stop growing any larger, our cells continue dividing as old cells die. Every day, there are about 2 trillion cell divisions in your body. So let's talk about how cells divide. This is a cell. You may know from your science classes that cells are full of stuff, like the endoplasmic reticulum, and the Golgi apparatus, and mitochondria. We're actually going to ignore most of that stuff right now, and just focus on the cell's DNA, its genetic material. Before a cell divides, its DNA is replicated and condensed into chromosomes, so that the dividing cell starts out with 2 sets of chromosomes. In order for the cell to continue growing, these chromosomes have to be separated accurately, with each daughter cell, here we have our 2 daughter cells, receiving 1 of each pair of chromosomes. This is no easy feat. Cells are extremely crowded places, and just one mistake can be lethal to a cell. Luckily, cells have an organelle that's up to this daunting task, the mitotic spindle. The spindle is a football-shaped molecular machine that divides a cell's chromosomes into 2 daughter cells. Unlike most organelles that are always present in cells, the spindle only exists during cell division. It assembles early in cell division, attaches to chromosomes, and moves them to opposite ends of the cell, and then disassembles again at the end of cell division. You may be wondering, how do we know all of this? Well, often it's because we can literally see it happening, like in the video that was playing earlier. That was a video taken with a technique called fluorescence microscopy. Microscopy means that we're looking at the cells through a microscope, an extreme version of a magnifying glass. This particular microscope makes things appear 10,000 times larger than they actually are. That lets us see things as small as cells. In order to see things inside the cells, we add fluorescent markers. Fluorescence is a special property some molecules have, where when you shine the right type of light on them, they shine light back, kind of like a glow-in-the-dark star. We can take these special molecules, called fluorescent markers, and attach them to other molecules inside the cell that we want to look at. That's fluorescence microscopy. What I've done in this video here is attach one color of fluorescent marker to proteins in the spindle, and another color of fluorescent marker to chromosomes. This way, we can see the spindle and chromosomes in real time as they divide. Of course, there's lots of other things in the cell as well, like the ER and Golgi and mitochondria, but since they don't have any fluorescent markers attached to them, they just look black and we can't really see them. The ability to watch molecules in cells with fluorescence microscopy is a fairly recent development in the last 50 to 60 years, and it's made a big difference for biologists, including those studying the spindle. We've learned so much from it. We know the shape of the spindle, its structure and speed, and a huge number of the proteins that form its structure and control it. But there are still limits to what you can learn just by watching something. One big category of things we still don't know about the spindle is its physical properties. Is the spindle hard or soft? Is it more like a liquid or more like a solid? Does it pull chromosomes apart from the outside or push them apart from the inside? In either case, does it have to use a lot of force or only a little force? Recently, I've been working on experiments to answer one small piece of these big questions. What does the spindle feel like? How hard or soft is it? How squishy is it? If the spindle were a larger object, we could easily answer this question just by poking it with our finger. We can easily take a pillow, feel it with our hands, poke it with our fingers, and tell what it feels like. We can tell, for example, that this pillow is softer than this table. A spindle-sized object, however, is way too small to poke with our hands, so we need a different tool. The tool I use is called a magnetic tweezer, and I use it along with fluorescence microscopy. You may remember playing with toys like this when you were a child, where you have a magnetic stick and magnetic balls. The magnetic stick you can use to move the balls around. The magnetic tweezer works exactly the same way. It just looks a little different. Let's head to the lab so we can see the magnetic tweezer that I use. Welcome to the lab. Here I'm in one of our microscope rooms where we have the magnetic tweezer set up on a microscope. This right here is the magnetic stick that I use to move the beads around. The beads I use with the magnetic tweezer are much smaller than the beads we saw in that toy. These beads are so small that their diameter is about 100 times smaller than the width of a human hair. This means they're so small that they can enter cells.
It's time to prepare the cells we'll do experiments on today. For my experiments, I use human cells that have been modified to be able to grow in a dish. Here, I'm taking the dish where cells have been growing in a warm incubator and moving some of them over to another dish that will fit on the microscope. I'm also adding beads to the dish, and after a couple days that are conveniently skipped over in this video, the beads will enter the cells and I can use them for my experiments.
So the cells are in this dish, which you saw me prepare earlier, and the beads are added to the cells. And then I'll position the tweezer to be right next to the cells. In order to see what I'm doing, I have the tweezer, cells, and beads set up on a microscope. I also have fluorescent markers attached to the spindle, to chromosomes, and to beads so that I can see them. Let's take a look at what I'm seeing. So this right here is what is coming out of the microscope through the camera that's attached to it. In magenta here we have markers for the spindle, in green we have the chromosomes, and in cyan we have the bead.
What I'm looking at today is how much the bead moves when I pull it through the spindle with the tweezer. If it moves a lot, that means the spindle is easy to move through, like water. If it doesn't move as much, that means the spindle is harder to move through, more viscous, like molasses or wet cement.
What's really exciting about science is that every answered question leads to more unanswered questions. With the experiments I'm doing, I'll hopefully add to the growing body of knowledge on the physical properties of cells. But there will always be more to learn, and more experiments for me or for you to do.